please join me in welcoming our next panel.
All remember how our game is played from yesterday. I do want to remind you, as yesterday, that if you have a burning question about 505B2, this is not the venue for those questions. That should be addressed somewhere else. But again, any of the topics that were just covered, the presentations, if you have any questions on those, please line up at the room. Do try to keep it, well, no, keep it to one question per turn. If you have additional questions, you can go back to the end of the line. But we want to make sure everybody gets a chance to talk. Those of you online who have questions, we know you can't say your questions, but you've been typing them into the Q&A pod as we've been going along. Please continue to do so. We've got moderators in the back of the room that are gathering those. Time permitting, we will work those into the mix. With that said, let me go ahead and turn it over to microphone number 1, please.
Thank you. Good morning. This was nice. This was a very interesting discussion. Confusing, maybe, a little from somebody from my point of view. I've worked in both pharmaceuticals and medical devices, and I'm glad to see Ellen Stevens on the panel because it'll give some balance to this. I didn't hear one single discussion on human factors or usability. And with all the discussion on issues that said that you suggest for errors in some of these combination devices, all of them are taken care of in human factors, human and use. Do you not apply the device component to this, or is this outside of a combination application like this? It's not what I expected to hear. Thank you.
Thanks a lot for that question. So what you did here is we were talking about the user interface in terms of substitutability for the product as compared to its reference product. That is it's separate from all the other issues that you just raised. Yes, those things are still applicable, but we were focusing mainly on the substitutability of the ANDA product to its reference-listed drug product.
But that's my question. If I have a device that functions similarly but doesn't look identical to the reference device, I have all my standards, I have everything in place, I've done biocompatibility, I've done all my human factors, and it does operate in the same fashion. No, it's not pink, it's green, and I've already done my human factors analysis, and I've asked the question, does this create a problem? From what I'm hearing, that it is now it's a generic, not just a generic drug, but it's now a generic device, and I have to look, feel, and taste like the reference device. That's not what I would expect to hear. I'd expect to hear that if I can prove up through my human factors and my usability, I have created a device that's equal.
So I definitely agree with all that. I think this goes back to the point that with a generic product needs to be considered the same as the reference product, and therefore we have to meet certain criteria that make it same. And so if for example, you designed a product and it functioned the same as the reference product, it followed the same critical tasks as the reference product does, and it had the same external critical design attributes as the reference product, I would think that that would be a very good indication that your product would be substitutable when we look at that user interface piece. Alan, do you want to add?
Yeah, Kim, I think you covered it pretty well, but I think the only thing I would say from a CDER standpoint, when we're looking at design validation of a device combination product, and I think the presentations covered it really well, what we're looking at in terms of substitutability is, are any of those differences going to impact safety or effectiveness as it relates to substitutability? You may be able to do a standalone human factor study and validate the design of a device, but that's not the question that's being asked. We're looking at substitutability of that product. So that's the nuance. I mean, it's a different category of product when we're talking about combination products.
All right, let's take our first online question. Those of you here in the room, if you have questions, come on up to the microphones, but let's go online, please.
Okay, our first question is, can acceptable design differences for one ANDA be considered acceptable for another ANDA with a different reference product?
Thank you for the question. I think that this is a very tricky question. If the user interface of two different reference listed drugs are exactly the same, then that question, if you made an ANDA to the first one that met all those criteria, that was found to be substitutable for that product, there is the potential that it would be acceptable for the second. But again, we look at it on a case-by-case basis for each of the RLDs. So you can't just refer to another RLD and say that the product was acceptable for RLD number 1, but I'm trying to make a generic to RLD number 2. So we would have to look at it on a case-by-case basis, do the comparative analyses for the specific RLD that you're referencing in order to make that determination.
This just goes towards the basic concept of that each generic product relies on a single RLD and not multiple reference listed drugs.
Okay, microphone number 1, nice and close, please.
Good morning. Thank you very much for the presentation. I have just one question, actually.
That was close. That was really close.
Absolutely. In one of the presentation, it says that you only need 2 batches of API to manufacture 3 primary XB batches. And it's not necessarily, it's not essential to use different batch of packaging material. And also in another presentation, I heard that the material difference, device material difference is a minor difference. To me, this is actually quite surprising. Normally, you need 3 batches of API, and you need a multiple batch of packaging material because different batch of packaging material could have different physical chemical properties. And the material difference could be significant. So can you please elaborate more on that? What's the scientific rationale that you only need 2 batches of API for 3 XB batches, and you don't need to use different batch of packaging material for the 3 XB batches?
Try to address your questions, then my colleague from CDRH can add it on from device perspective. So just try to clarify. So one of the slides Peter present is about the requirement, how many batches you need. Then you mentioned 3 batch API. I think it's 2 batch API, right? You
yes,
need 2
exactly.
API, not 3. But we normally require 3 batches. So we always request 2 API for supporting these 3 batches. That's normal. But then we probably, Peter mentioned, for components of the container, say you have auto-injector, in worst case, most complicated case, you probably only need 1 batch. Consider CDRH has reviewed that device, find acceptable. So you don't need to treat that device part like API. That's our current thinking, okay? We don't need multiple batches of those container closure. Just like anything else, we don't require you multiple lots of container closure to support your ANDA. So same principle apply here, assuming the device was found acceptable. So if we're still not, also assuming that device parts has no significant impact for the drug product performance, right? So if you're still not sure about this, you can submit a controls and to make sure. But I'd like Alan to add it on the device from device perspective to the requirement. How many batch you want?
Yeah, I don't have too much to add. I would refer you to slide 28 from Beta's presentation, where she discusses the requirements. I mean, we are looking from a device stability standpoint. We'll be looking at least 3 different batches of data to be put on stability. But I would refer you to the slide she discusses that information.
Thank you.
Microphone number 2, please.
Good morning. It was a nice presentation about this combination product. So one of the thing which is missing is a transdermal, which is invariably we receive deficiencies related to the combination product questions for the transdermal product, transdermal patches. So is transdermal patch is a combination product or is it just a drug product?
Thanks for the question. Yes, so transdermal patches are considered combination products, and we actually covered those yesterday. So if you have some questions about transdermal specifically, you can come and catch one of us later in the presentation. Or if you have a specific question with regard to your product that you may be developing, that would be, you could submit a controlled correspondence to get an answer to that as well. Thank you.
So the lots used, because we are discussing about the number of lots and all the APIs being, there are like almost 15 components, like the release liner, backing membrane, you know, the various excipients used in that one. So the challenge is like 3 different lots of the every excipient used in manufacturing of the transdermal product. Is that mandatory, or if we show that there are not much difference between the lot 1, 2, 3 of the every excipient and show the equivalency, would that be allow us to use one particular lot for this to manufacture 3 batches of the transdermal product?
Yeah, so we have a session yesterday talking about whole requirement for transdermal, and I'm sure we have people in the audience, maybe from the agency, can help you answer these questions. But this session, I want to focus our discussion on the injectable products. Is that okay? I mean, we can talk about after session. You can see me, or I can introduce you someone to answer your questions. Is that okay? Thank you.
And a reminder, all of the recordings from yesterday's session, if there's anybody here that had to step out and miss some of those, are available online now. So you can go back and review those during the lunch break and then perhaps get some answers there. Let's actually go to an online question, please, and then we'll be right back.
Okay. The question is, why is 99.999% reliability required for an emergency auto-injector?
Yeah, thanks for that question. So from the device standpoint, you know, when we're looking at the functional performance of the device, one of the things that we are considering is the reliability of that product. So if you look at the injector guidance that was referenced in Peter's presentation, you know, one of the performances that we're looking at is the consistent, reliable performance of that product. When we talk about emergency use, obviously that's a situation where we need the reliability of that product to be adequate, to provide adequate assurance that it's going to function as needed. And so the actual specification that was listed is something that we're asking for certain types of products. And that's something that the agency has been working on guidance. So that should be, you know, hopefully, I can't obviously discuss when that would be released, but that's something that the agency has been public about, that we're working on putting that additional details out.
Okay, microphone number 1, please.
Good morning, and thank you for all informative presentations. It was wonderful. My question is mainly on the combination product, where the product is given as a kit. It's a lyophilized vial, and the diluent is in pre-filled syringe. So the same diluent is added into the vial, reconstitute, and in the same syringe you withdraw the product. So the question is related to mainly quality. To what EPR? We have to do the EPR of mainly like light force, breakout force on the diluent or on after reconstitution.
Yeah, thanks for that question. So when we're looking at device performance, we would be looking at, as the product is being administered, we would want to see those EPRs being assessed at that point. So if the actual reconstituted product is going to have a difference.
But the diluent is already, I mean, the breakout is already over because you are already transferred to the vial. So again, doing in the same syringe doesn't make any sense.
Right, yeah, so you're reconstituting and using the same syringe to administer. Yeah, so we would want that testing to be done as it would be administered to the patient.
Thank you.
Microphone number 2, please.
Good morning. My question is regarding healthcare administered products. In one of your presentation, you said that there are some differences which are allowed differences for healthcare administered products. Can you give some examples of that?
Thanks for that question. So yes, there are considerations when we're doing that comparative analysis and we're looking at that user interface piece. We do take into consideration who that user group is. So Commander Fine came up and showed you some and gave you some information about, for example, if there's if the needle cover has that automatic lock, for example, if that were given to a patient, an end user. So patients don't go to medical school, they don't go to pharmacy school, they don't have a lot of advanced training in most cases, and so for them, having a safety shield on a needle could be very confusing. And the risk is that if you accidentally bump that needle cover on in your haste, you've now locked your syringe and you can't get your potentially life-saving dose of whatever it is that was in there. So that could be a terrible conclusion. You could have a patient that actually dies because of that user interface difference. So, however, if the end user is only healthcare professionals, the expectation is that healthcare professionals have experience with a wide range of device and user interfaces. They are used to going from one syringe to another syringe. They're used to reconstituting products. They're used to using a multiple range of devices by which to deliver certain drugs. So that user interface for a healthcare professional only indication, we would be much more liberal with what could potentially be substitutable because we know that that end user has a vast array of experience on which they rely to be able to deliver that product.
All right. Online question, please.
Okay. For an ANDA that proposes a pre-filled syringe where the reference listed drug is in a vial, does that change normally require a human factor study for approval?
Thanks. So the pre-filled syringe versus a vial has been something that FDA has historically allowed as an acceptable change for a device. If it's considered an acceptable difference in a product, then the difference is acceptable, and therefore we would not expect a comparative use human factor study. Let me go back a little bit because it seems like there's a lot of confusion about comparative use human factor studies and when you need or don't need them. So first, what we're talking about, what the guidance proposes, is that you do comparative analyses, which are those 3 steps to look at the differences between the user interface of your product and the user interface of the RLD on which you're relying. So once you've made those 3 steps and you've looked at those, if you find no differences or if you find only minor differences, meaning that the differences do not affect an external critical design attribute, then you are essentially, you put that information in your ANDA and you say, we found only minor differences, here's what we did, here's what we think about it, and if you want to, you can even include your risk evaluation that goes along with that thought process, submit that to your ANDA, and you're done. You do not have to do any additional information or data to support that application. If there's a difference that may affect an external critical design attribute, and you are not sure, or we are not sure as FDA, then there are cases where we will ask for additional information or data. One type of additional information or data is what we've termed a comparative use human factor study. Okay? There are many other kinds of information we could ask, many other kinds of data. So an example would be, I'll talk about this a little later this morning, so I'm going to jump into an area I'm more comfortable with for just a moment, so bear with me. If you have an inhaler device and there's a slight difference in resistance between your device and the reference product, you might be able to give us some in vitro data that is supportive of that potential difference in resistance that may be found acceptable because we're not sure if that's going to impact or not. So That's the kind of information that's not a comparative use human factor study that could also be used in a situation like that where you're looking at a comparative difference that may be in that other than minor category. The final thing that I want to add is that if you have a difference that it definitely impacts a critical task, or your critical task is different on your test product versus the reference listed drug, or you have an external design attribute that's totally different. For example, an auto-injector that has an extra button that you have to push as compared to the RLD, which just maybe swing and stab. If you have something that you know is different, there's a high likelihood that that would not be found acceptable for the user interface difference, that would be too different. You couldn't be considered the same because the critical tasks and the external critical design attributes are too different for that product to be substituted at the pharmacy level and not require additional training to the patients or to the end users. Okay, so hopefully that helps clarify just a little.
All right, microphone number 2, please.
In continuation to the online question regarding the reliability of 99.99% for auto-injector, for a simple PFS syringe, although used in emergency use, is 99.99% reliability or 99% reliability is required? How do we decide?
Can you restate your question?
How do we decide the level of reliability for PFS syringe used in emergency use?
How do you decide how adequate
Level of reliability.
Yeah, I mean, we haven't really established from a syringe standpoint, what would be acceptable. We're looking mostly at devices that have automated functionalities when you can't observe whether or not the device, until you actually attempt to use it, whether or not it's been assembled appropriately and it's going to function. So syringes, it's a different level of risk. It's a different category. So I think, for now, we're relying on manufacturers in that space to define what they would deem as acceptable levels of reliability, and we would have those conversations during review. But that's sort of the process that we use. I would refer you to ISO 14971, which is a risk management standard for medical devices, and it discusses in there. It has examples of levels of risk depending on levels of severity, and so that's one of the things that we've relied on when we're doing our reviews and making these decisions.
Microphone number 1, please.
So as per ISO, Class B devices require one site specification. And especially my question is on EPR, where you have delivered dose volume measurement. If the product is a suspension, so when you say volume measurement, instead of that, if we can do the content analysis, what is delivered, would that make acceptable?
I didn't catch the end of your question.
So for Class B devices, this only one site specification is applicable. So if you, on reconstitution, when you delivered, the requirement as the EPR requirement is, deliver dose volume. Instead of measuring volume, if you can do the content analysis, since it is a suspension, so especially where the particles settles quickly.
Gotcha. Yeah, so it sounds like there's different things going on for your product. So if we're looking at a device that's delivering a drug, and the requirements of that device are to deliver a certain volume per time or per injection, or whatever that specific device is. From the engineering standpoint, we're looking to see that you have evidence to verify and validate that the device is doing what you intended to do. Whether the suspension is acting appropriately or not is a drug quality issue. That would not be something that we would, in CDER, would be addressing.
Thank you.
Online question, please, Lisa.
Okay. How should specification criteria be set for dose accuracy for pre-filled syringes? Should criteria of no less than the nominal volume be followed, or is there an upper and lower limit around the target value that's acceptable?
Understand questions about the dose accuracy. So so you have to meet USP1. I guess you have to make sure you can deliver label claim contents If it's label says to deliver 1 milliliter, then you have to deliver 1 milliliter. I don't think we have acceptance criteria, upper or low limit. It's just 1 milliliter. You have the prefill syringe, or you have the device, or tell you exactly volumes. So yeah.
Microphone number 1, you can tilt that down a little bit if you want.
Hello, thank you for the presentations. I learned a lot from it. And then actually, my question is still regarding the overfill for the long-acting drug, which is we developed the Institute implant formulations. Typically, the overfill for the preload syringe regarding of the amount of the drug we load in the syringe is quite high. It's way higher than the USP standards. So the formulation actually does not involve the patient reconstitutions. It's the preload ready to fill. So when we prepare for the filing, the packages, so what's the guidelines we should follow for the overfill part of the regulatory, the document preparation?
And this is very interesting. So yeah, we didn't discuss about this today in Vita's presentations, but yeah. So normally we have this slide. So for prefill syringe, for regularly for injector product, you have USP 1151 to
1151.
Recommendation depends on the product. It's suspension or it's solutions or it's aqueous solutions. You have added including extra amount to ensure the delivery.
Right.
But so for free syringe, it depends on your labeling, right?
Yeah.
So it's a little tricky. And the prefill syringe, there's some dead space in there. So you have all considered that. So you have to provide your product development. Say, I want to include certain overfill.
Right.
So this overfill can compensate those dead space, as well as sometimes the label says, oh, the patient giver need care, either, like to plunge this, prime your syringe a little bit, couple drops. So you have to consider all this.
Right.
And then you can propose your overfill for a prefill syringe.
Okay. So there's no, the limitations, how much, let's say, for our case, typically we have to overfill way a lot compared to, because they are a high potency drug, so the injection amount, which is the patient received, compared to the overfill is small. So the ratio is actually way higher than the USP standards. So we were wondering is for this high potency drug and that's the amount need to be injected to the patient received amount compared to that.
So your good question. So there is something, I think we have a guidance about excess volumes for injectable product.
Right.
So the only thing is you don't want to put too much in there, cause medical error, because someone, if they don't follow label instruction, then patient end up with excess drugs. Also, sometimes we try to avoid, if you put it, say, 50% more, so you presumably you can give like one dose and a combination dose. I mean, that's not probably for prefill syringe, but for vial, you can actually label says only 5. It's 5. So if it's multiple dose, it's 5 dose. You probably can give 6 dose. So we try to avoid that. So I recommend you refer to our guidance for excess volume for injectable product.
That's my question. Typically, if we follow the current guidances from USPs, basically that will be much lower. So typically for our case, we load into the small amount is high potency drug compared to is preload syringe. The patient does not have any involvement with, as far as you just one push, and then as far as you push all of them in, they will receive the correct dose. But for overfill, we have to overfill way more than the actually USP standard 115 gave the recommendation.
Yeah, sounds reasonable, but if you're not sure exactly what we're doing, sounds your case is a little complicated. I will recommend you submit a control correspondence to ensure your proposal is acceptable.
Oh, okay.
Thank you.
Thank you.
And again, we still got some time, so if you've got questions that aren't really specific to your product, but of general nature on what was discussed, please come on up to the microphone. Before we go to the mic, though, I'm going to take another online question.
Okay, we had a couple people ask this question. Can you explain why drugs or dilutants packaged in flexible IV bags, why they're considered to be a combination product? And additionally, how can people find out if their product is or is not a combination product?
If you look at the regulations, and I don't have the one in front of me, it's somewhere in the 800 regulations, it defines an IV bag as being a device. I'll also, if you want to know whether something is a combination product, remember that there's always going to be two different constituents, right? So you're going to have either a constituent and a device constituent, or a drug, the microphone that works. Or a drug and biologic, or a drug, biologic, and device. If you have specific questions, you can submit a RFD, which I mentioned in my presentation, or a pre-RFD to the Office of Combination Products. That's the office within FDA that has the jurisdiction for determining whether something is or is not a combo product. And the other thing that I'll note is we've had a number of applicants come to us and say, look at the Daily Med website. It says that this is or is not a combo product, and I'll just note that that is not populated by FDA, and so that may or may not be accurate.
I'll follow up on that. My name is Steve. I'm the OPF individual on the list. Like Lisa said, for devices, I think it's 860s through 880s describe lots of devices through general hospital devices or IV bags or dental devices. So you can see explicitly what devices are there and listed as medical devices, and also what classes they are. So if they're class 2 products, class 1 products, so the risks and the level of complexity that they have there.
You can't leave yet. Microphone number 1, please.
Hi, thank you. My question is more about the process. I'm new to combination products, and you talk about the primary mode of action. Oh, you go to CDRH, and why, if my product is assigned to CDRH, then they don't care at all about the API, and I can do my 510K and get approved, and then I start claiming some things, and the RFD says, oh no, now you foresee there, and the product is the same, but suddenly I became a drug, and now the API is all important.
Lisa, you were just given a microphone.
So I am not a scientist, so I can't speak to the API piece, but I'll note that we do what we call a substantially similar evaluation, regardless of which center it goes to. And so if we have a drug-led combination product and it comes to CDER, we're going to do a substantially similar evaluation if that had been actually submitted to, that device constituent had been submitted to CDRH. I can't, though, speak to the exact part of the API question, but I hope that helps. I'll also note that you can refer to, it's called, I want to say it's our pre-market expectations guidance for combination products, and that will help answer some of these questions about primary mode of action and how we conduct our reviews for these combination products.
Which guidance you mentioned?
I want to say that it's, these names of our guidances are so long. I want to say if you, at least, I go to Google, pre-market expectations for combination products, that should help pull it up for you.
Alan, did you want to add something?
To add on real quick, if you go to the Office of Combination Product website and their guidance list, they have, I think might be listed there, has GMPs, pre-market product guidance, and lots of other ones there. So that's probably an easier way to find it.
Yeah, and just to add on to what Lisa said, it sounds like you have a very specific situation, and obviously I can't speak to, I don't know what it is, but just from a process standpoint, when there's a device-led combo or a drug-led combo, the centers are working together to make sure that those submissions are reviewed appropriately. And so if there was a device-led combo that didn't ask some questions, and then later on you got asked other questions when it was a drug, there probably were reasons that were behind the scenes that the centers worked out. But in terms of process, that's how we do our work. When we have a device or a drug-led combo, the centers are working together to make sure those are reviewed appropriately.
Okay, microphone number 1, please.
Thank you for the excellent presentations. For the pre-filled syringes, does the container closure integrity testing have to be conducted over the stability period?
You repeat your question.
Does the container closure integrity testing for the pre-filled syringes have to be conducted over the stability testing at different stability time points as well?
Yeah, so when we're looking at a prefilled syringe, we would be looking at the device performance aspect of that on stability. Is that what your question is?
Yes.
Yes.
Okay, at different stability test points, the integrity testing have to be established.
Yes, that's correct.
Okay, thank you.
Do we have another online question, please?
Do we need to use the to-be marketed combination product to conduct our comparative analyses, or can we use a product that's still under development?
Thank you. This is a great question. So what I would suggest is yes and yes and yes. And what I mean by that is that when you are in development, you should be thinking about doing comparative analyses to look at that user interface piece throughout your product development. If you wait until the very end, you may miss the fact that you had an other than minor difference that now you have to go back and redesign all the way up from the beginning again. And if you only look at it once in the beginning, but then make multiple changes throughout your development process, you may also miss something that comes through. So what I would suggest in that case is that we have a process by which you can submit a controlled correspondence for your device user interface assessment to the Office of Generic Drugs. You can submit to us your comparative analyses. We will take a look at them, and we will provide you information back in a controlled correspondence. So there's a 60-day clock on those device user interface assessments. So you can actually get answers back within 60 days, which for the FDA is kind of fast. So that's one way that you can have an assessment of that. Another way, and you can do multiple, you can submit multiple controls over the course of your development program. We don't want 5 in the same week. But if you submit it and then you make a change to your product, say you had to make a small device change based on something else that you were doing with the combination product, you could then go and submit another controlled correspondence for your now modified device, and we would continue to work with you in that pre-ANDA space to help you with those assessments. And finally, if you have multiple questions for your package, you might also be eligible for a pre-ANDA meeting if you are a complex combination product as well, and that's another place where we would also be able to provide feedback on those comparative analyses assessments.
Okay, another online question, please, Lisa.
Okay. If we have a combination product with a class 1 device, for example, films or oral syringes, do we have to prepare a separate device history file and comply with that Part 820, that 30 design control requirement?
I think if you look at Part 4 and if you look at the CGMP guidance for combination products, it explains the agency thinking on what the quality system requirements are for the device constituent. And in general, we're trying to align between what the device requirements would be if it was a standalone product as it is when it's a combination product to the extent possible. So, you gave the example of an oral syringe, and that would generally be, I think that was one of the examples, or some other types of liquid medication dispensers are examples provided in the CGMP guidance for combination products. And so I think if you look at that guidance and you look at the individual regulation for the device that you're working on, that would be the place to start when you're trying to figure that out.
I'll follow up on it. So class 1 exemptions for life system regulations like you're describing, it's talked about in 21 CFR Part 4, which is GMPs for combo products. I think there's some caveats where if you have a class 1 device within a combination product, the intended use needs to be the same, the labeling needs to be the same or similar. So if you're taking a class 1 device but you're using it in a way that's not being used by itself or intended as by itself, then these exemptions probably be appropriate for this product. But I think it's in the guidance or within the regulation. But summary that Class 1 exemptions do exist or described within either the guidance. There's an alternative mechanisms for GMPs federal notice that was published last year. So I think that has more information about Class 1 exemptions for combos.
And 24 seconds left. One more question, as long as it's nice and quick. Go ahead.
Okay, about design history file, and I understand the need to develop design history file. We develop our device in accordance with the reference device. So we will not go through many of the development phases as a reference product. Therefore, our design history file will be very different than the reference design history file. So my question to the agency is that, what's your expectations for the design history file for a substitutable device?
So I think, from the CDER point of view, when you're talking about design controls for a combination product, generic combination product, you're still responsible for documenting what the design inputs and outputs are for your product, how you verified and validated that your product is meeting its requirements. What we're generally considering in terms of substitutability, comparability, is when we talk about design validation, are those requirements adequate? How did you develop those requirements? You're really relying on the reference product for validation of a lot of those performance requirements. But you still have to do a lot of design control work. And so that design history file is going to include the information specific to your product.
Short add-on, Alan can correct me if I'm incorrect, but for design controls, it's more about the method of performing design activities within a product versus like having the exact design of a reference product. So for your product, if you're doing design work and it's the way you do it with the, incorporating inputs and outputs and verification and validation of that design, that's kind of what design control is about.
Yeah, that's a good point. So I went right to the product aspect of the design. So the design control processes that you implement are specific to your product, and then everything else that I discussed about the actual design outputs and how you've documented those are also specific to your product. So it's both process and product that need to be addressed.
Thank you.
All right, and that is all the time we have for this panel. Please help me thank the panelists and this morning's speakers. And this morning's speakers were kind enough to actually get us a little bit ahead of schedule.